Welcome to this international conference of the National Institute of Genomic Medicine. Today, we have as our guest speaker Dr. Katherine J. Franz from Duke University. Dr. Franz attended West Wellesley College and graduated in 1995. She moved to the Massachusetts Institute of Technology for graduate studies, where she worked under the supervision of Stephen Lippard and completed her PhD in 2001. Currently, Dr. Franz is the chair of the Department of Chemistry at Duke University. Dr. Franz's group's research focuses on elucidating the structural and functional consequence of metal-ion coordination in biological systems. She is particularly interested in understanding the coordination chemistry used by biology to manage essentials yet toxic species such as copper and iron. Understanding these principles guides our development of new chemical tools to manipulate the localization, speciation, and reactivity of biological metals ions for potential therapeutic benefits. Today, Dr. Franz will present a lecture entitled Infectiously Inorganic, a metallocentric view of antimicrobial activity. Let me say the following about this conference. Normal and pathogenic cells require a menu of metal nutrients for optimal growth, but also strategies to mitigate toxicity associated with misregulated or excessive levels of metals like iron, copper, and zinc. Cells adjust metal homeostasis mechanisms depending on cell type, local growth conditions, and in response to stress. These situations present opportunities to manipulate cellular metals as a therapeutic strategy across a number of diseases. Here, she will present a metallocentric view on utilizing small molecules and peptides that leverage unique methallobiology associated with bacterial and fungal infections to selectively inhibit growth of pathogenic microorganisms. More broadly, this approach is used to explore how cellular response at the metallomic level affects and are affected by microbial susceptibility and adaptation to antimicrobial treatment. Dr. Franz, welcome to the National Institute of Genomic Medicine.
Thank you very much for the introduction, and it's a pleasure to be with you here virtually and tell you about work that we're doing in my laboratory in North Carolina at Duke University. So I want to start by acknowledging the students and postdocs who've contributed to this work. This is a current view of the group, but actually the work I will tell you about is mainly two former graduate students, Abigail Jackson and Jacqueline Zengel-Baroni. And I'll also tell you a really nice story. It's a collaborative work with my colleague, Professor Michael Fitzgerald, and his just recently graduated PhD student, Nancy Corsi. So I'd like to, I know we're at a Genomics Institute, but I'd like for you to think for a minute about the metal cations that are in our bodies and in our cells and how they get to where they need to go. So metals like the ones that are shown, we know that we need them for important biological processes. And just some examples include the iron that is needed in our hemoglobin to carry oxygen. Let's see. Something's, hold on a second. I'm going to restart this. Okay, something was frozen. So other examples include iron and copper that's required in cytochrome C oxidase. So this is a very life important reaction that's required for respiration to take oxygen to water, and it requires both of those metal cations to be in their appropriate places. And if another example are zinc transcription factors, and the binding of zinc is required to hold those stabilize those proteins so that they can recognize DNA. So these are all life essential processes carried out by these sometimes called trace metal cations. But in biology, this is a very intentional process to get these metal cations across cell membranes and into either carrier proteins or chaperone proteins that can eventually get them to their requisite enzyme or protein active site. And metal sensors that can detect if there's too much metal around. So these metal cations usually come in through dedicated importers or transporters and can be expunged from the cell if in excess with dedicated export proteins. So we think about that balance of having just the right amount of metal to carry out essential function without having toxicity as metal homeostasis. And so you think of a comfort zone that's somewhere between not enough, which would lead to a starvation response, and too much that can lead to a metal-dependent toxic response. And what we like to think about in our lab, we're a bioinorganic chemistry lab, is how we can use chemicals and what we know about metal binding compounds to affect this comfort zone, maybe push cells outside of their comfort zone by inducing toxicity or preventing some kind of essential process in a disease state. For today's talk, we'll be talking about antimicrobials. So pushing these compounds pushing these cells out of their metal homeostasis zones or their metal comfort zones. So this is true for all cells, but now take these cells and put them in this interface between a host and the microbes that live within and on us that can become pathogenic or when we acquire different pathogens. And so each of these different cell types needs this metal homeostasis or its own metal comfort zone. And when we think about pathogens now, we're thinking about this battleground over the metal cations that they have to acquire from the host or protect themselves against host-induced metal toxicity. So there are many examples of a principle called nutritional immunity or metal sequestration, where there are processes in our immune systems that, for example, withhold iron, put us in an anemic state to prevent infection from spreading. On the flip side of that are examples where our immune cells actually use bursts of copper and maybe even zinc as a strategy to try to clear infections or to kill those pathogens. That's from the host side. From the pathogen side, now these microbes are incredibly adaptable and they will change the way they import their metals or export their metals depending on the environment that's around them, in this case, a host. They can express and produce molecules that can help them acquire nutritional metals that they need, and they can even switch their metal cofactors. So for example, there's an enzyme called copper zinc superoxide dismutase that's very important for preventing oxidative stress. And at least in some yeast cells, under conditions where there's copper deficiency, they will switch from using their copper zinc SOD and instead use a manganese dependent SOD, just to be able to adapt to changing metal conditions. And what we do then is think about, okay, we have this battleground between host cells and pathogen cells over the need for metals and the protection against metal toxicity. What happens if we add a drug-like molecule or a small molecule into that mix? How does that affect the efficacy of that drug? From a chemical perspective, you can think of both a direct interaction between the drug molecule and a metal cation to form a metal complex that might change the way that metal moves around the cell or its targets. You might have a small molecule that can inhibit a metalloprotein as its target, and that will have a metal-dependent response. And then you can think of other conditions that might not bind the metal directly, but can involve a disruption in this metal homeostasis process or in this comfort zone. And today I want to tell you some brief examples that touch on all of these examples of using small molecules to push these pathogenic cells out of their comfort zones. So to start, I want to introduce two classes of metal binding molecules that you can think that could either push towards starvation or push towards toxicity. And over here in the bottom left are examples of metal binding sequestering agents that bind metals very tightly and inertly and kind of prevent the organism from accessing that metal payload. So these are things like siderophores, and this molecule is called BCS. It's a copper sequestering agent that we'll use later in the talk. The other hand are metal binding compounds that form lipophilic metal complexes that can actually cause an increase in metal concentration inside the cell. So they change the localization of metals and they can give up the metal. They might not be as tight binding as this other class. And we call these ionophores or ion-loving compounds. And at least in our hands in the examples we've looked at for a cytotoxic activity, we see that this ionophore activity of inducing a metal toxic response with these ionophores can be very effective. So highlighted in the center here are three examples of these ionophores that can induce a copper-dependent toxicity. They're old molecules. Pyrithione, for example, is the main ingredient in many dandruff shampoos. So if you used a dandruff shampoo this morning, you may have used a zinc pyrithione bioinorganic complex. So these are old molecules. And what we've thought about a lot in my lab is how we bring some new innovations to these molecules and make them selective for certain disease states. So for example, we introduced this molecule we call QBP that is non-toxic, but under conditions where a macrophage in our immune system senses a pathogen and induces an oxidative burst, that change in the oxidation environment converts this QBP to these hydroxyquinolines that now can induce metal-dependent toxicity. And we have examples in a cancer setting using these dithiocarbamates, again, in a pro-chelator kind of fashion, like a pro-drug fashion. And the one I'll tell you more about in an antimicrobial action is this molecule called PSEF-PT. And I'll tell you more about this molecule in the next slide. So before I tell you about the molecule, I want to focus your attention on this structure in this E. coli cell membrane. This is the peptidoglycan cross-linking that gives the cell a lot of stability. And I love these images from David Goodsell that remind us just how packed the cell is with all these structures. But this peptidoglycan layers give stability to the cell, and it's formed in one of the steps by this penicillin binding protein, or PDP, that cross-links this D-ala-D-ala substrate to make this strong layer. But our antibiotic compounds, one of the ways that they act as antibiotics is that you can see in red, it mimics the structure of the D-ala-D-ala substrate. And so these penicillin, cephalosporin, these beta-lactam antibiotics inhibit penicillin binding protein and prevent that cross-linking, making the cells then not stable, and these are toxic compounds. Of course, there's a great threat in drug-resistant microbes, and one mechanism that they acquire drug resistance is they acquire beta-lactamase enzymes that cleave these beta-lactam drugs in this four-membered ring. And now you can see that the cleaved compound doesn't have the same structure, and you don't get this inhibition, and then that leads to drug resistance. What we've done is taken a cephalosporin core and fused it with one of these ionophore compounds. So in this prochelator version, it does not bind metals very well, but when beta-lactamase enzymes are present that recognize the beta-lactam core and cleave it, so this would be under E. coli cells, for example, that have drug-resistant beta-lactamases. Now we produce this metal ionophore that can have metal-dependent activity in a way that takes advantage of the drug resistance itself. So here are some of the data that we've acquired. So to orient you on this particular experiment, we're plating out E. coli cells in 96 well plates, and we increase in concentrations of our added compound, and we're looking for the minimal inhibitory concentration where we stop seeing cell growth. And we're looking at three different conditions. One just in the cell growth media, and then one where we add a little bit of copper chloride. This is not enough copper to actually kill the cells, so it's a low concentration of copper. And we also have a condition where we add that copper sequestering agent, BCS, that I mentioned earlier. That creates a copper deficient environment because all of the copper gets sequestered in BCS. So here are what some of the data look like for a control compound. Cephalothin is a current antibiotic, beta-lactam antibiotic. And we have a couple of different E. coli cell lines. These are all in the same background. So here's the null cell. And then we have in the same background, these four different beta-lactamase enzymes that are being expressed. OXA-A1 is a penicillinase that doesn't cleave carbapenems or cephalosporins very well. And then TEM1, CTXM1, and CMY2 are all drug-resistant cephalosporins, and you can see that by we add cephalothin and we get no, we have to go to very high concentrations. We don't see any inhibitory concentrations, but we do a little bit with OXA-A1. If we look at the ionophore compound itself, I should mention that we don't see a difference in copper dependency, either copper or copper sequestration with the antibiotic itself, which we expect. Now with the metal ionophore PT, you can see its behavior is very different. Now these strains that are resistant to the cephalosporin are susceptible to these metal ionophores, and there is a metal dependence. So adding a little bit of copper actually helps, but sequestering copper decreases activity completely, and we see nothing, no inhibition. This tells us that the mechanism of action does require or depend on copper in some fashion. And then our prochelator molecule, you can see, is a kind of combination of these two responses. In the strains that do not cleave very well, the cephalosporins, our molecule looks like a cephalosporin. It has some activity. It's not great, but it has some activity. But in the strains that we expect cleavage of our prochelator, you can see that the activity mirrors that of pyrithione, as we would expect if this molecule gets released in these cells that have the beta-lactamase. So we go on to do another experiment where we do the same MIC experiment, but then under the conditions where we stop seeing growth, we plate that out onto fresh agar and then look to see if the compound causes a cytal response and there are no cells that grow, or if the compound just inhibited growth and in the absence of the compound, cells will grow back and we just get a static response. So these are the data that I showed you in the last slide in a different form as the growth profiles of the CTXM1 strain. And you can see above 20 micromolar, we get growth inhibition. And in the null strain, it's acting like our antibiotic and we get no cell growth as we would expect if it's a cephalosporin in a drug-sensitive strain. But what we see in the cases of the drug-resistant ones that we see some cleavage, we do get some regrowth, and PT under these conditions also we get some regrowth, indicating that it's a static drug. But if we supplement with a little bit of copper in the media, now we see an improvement in this activity, and then across the board we see cytotoxic activity, telling us that copper, again, is part of its mechanism of action and that we can improve the toxicity of it by adding a little bit of copper. And it's very specific for copper. We don't get the same response by other metal cations. Again, this is the same data that I just showed you with CTXM1, and I just want to highlight that CTXM1 is a serine beta-lactamase. So it does its hydrolysis chemistry by using the serine residue to do the hydrolysis. But there are also metal-dependent beta-lactamase enzymes. Here is an NDM1 enzyme, and you can see there are 2 zinc ions in the active site. And these enzymes use a hydroxide bound to the zinc in the active site to do the hydrolysis chemistry. And you see that in a strain that expresses NDM1, PT looks just the same, but our prochelator PSEF-PT, we never quite get all the way to growth inhibition. It kind of slows it down, but it doesn't inhibit it. And what Abby was able to figure out, the first question was, well, does NDM1 not recognize our prochelator very well? Does it not turn it over? And it does. Here's a UV-Vis experiment. PSEF-PT is in blue. And you can see if you add an enzyme, that spectrum in red matches the spectrum you expect for PT itself. So it does turn it over. She then did a dialysis experiment of the isolated NDM enzyme with a metal chelator, EDTA, which pulls zinc out of the active site and doesn't recover any zinc. And then with both PT or PSEF-PT, you can see at even high concentrations, zinc is still bound in the active site. And that's telling us we think that the molecule is acting as an inhibitor, as I mentioned at the very beginning, an inhibitor of that metal-dependent active site. And so knowing that information, Abby was then able to recover the meropenem susceptibility. So it's a busy table, but focus on meropenem is a carbapenem, and you can see a very high MIC in this NDM1 expressing strain because the strain is drug resistant. You can see that PSEF-PT, we also never quite get growth inhibition, but a combination of the two at low levels of meropenem, now we recover meropenem activity. We think because we're inhibiting the NDM active site. So we have a multifunctional activity of this prochelator called PSEF-PT, where we can restore meropenem activity by inhibiting the zinc active site of NDM1. But more generally, we go back to the idea that this released pyrithione in the presence of copper can induce copper toxicity. And at this point, we're very puzzled. We're very puzzled about how it actually works. So let me give you a sense of what we think is happening. So we start with the copper-2 complex of pyrithione. We think it enters the cell probably through some metal transporter, very generic metal transporter, not specific, but we know that it gets in the cell because we know that copper gets into the cell. So we get an increase in copper and with pyrithione. We start as copper-2. We think that inside the cell, it would be released and reduced as copper-1. And the data that supports that are a whole cell EPR experiment where EPR can measure unpaired electrons. So copper-2 has an unpaired electron that is EPR active, whereas copper-1 is EPR silent. So the cells as isolated give us no spectrum. They're in here in the baseline. But if we oxidize them, then we can see this copper-2 grow in as to tell us that that latent copper-1 was in our cells. It just to see it, we have to oxidize it. So we know that it's copper-1. And that these other transporters are here to remind you some of how E. coli protects itself against metal toxicity, specifically against copper toxicity. One, it expresses this protein called COP-A. It's an exporter, and it will efflux copper outside of the cytosol into the periplasmic space, where it can be oxidized by QEO to copper-2. There's also a Cus system that can also recognize copper and get it out of the cell. Okay, so our question, and what I want to focus for the rest of the time of the talk, is we're very puzzled by how we could figure out what are the cellular targets of this extra copper. We know that we're getting a toxic response, but why? What's actually shutting down? So a key here, well, two things. We wanted to know where the copper was going, but also we have this small molecule now in the cell, and we know it can have effects. We know it can bind to metalloenzymes, for example, and it can probably bind to others. So we were very interested in what the targets are of this combination of the copper and the small molecule, and how are we going to figure that out? And here's where this really critical collaboration with my colleague, Michael Fitzgerald, who's a proteomic mass spectrometrist, and his student, Nancy Corsi. And this is where that collaboration really comes into play. And to go back to some background, thinking about protein folding and protein folding stability, this becomes the key to answering how we're going to answer our question about the targets of copper and pyrithione toxicity. So a protein is in equilibrium between its folded and unfolded state. And we know then so there's an equilibrium constant associated with that. And we can get from that a delta G of folding. So this gives us a measure of the thermodynamic stability in this protein folding process. And buried within that delta G, that can come from what would cause a change in that thermodynamic stability, protein misfolding, maybe point mutations, maybe post-translational modifications. And the one we'll focus on for today are binding interactions with some kind of cellular ligand, where that ligand could be for example, metal cation. So the binding of generically a ligand, in our case, metal, the binding of that, which has an equilibrium associated with it, will stabilize the folded form of the protein. So we get a delta G of stabilization. So if we can measure this stabilization change, that may give us a way to infer changes in protein function as a consequence of these different conditions that we treat ourselves with. So how do we measure this change in protein stability? Well, you can induce protein unfolding by exposing your folded protein to an increase in concentration of denaturant, like urea or guanidinium chloride. So that will shift the equilibrium from the nicely folded state to the unfolded state. And if you have a way that you can measure the fraction unfolded, or the fraction of folded protein as a function of denaturant concentration, you'll get a nice curve. And here you'll get where the C1/2 of that curve will lead you to be able to calculate the change in thermodynamic stability, or the delta G of folding. You can do this there are a number of methods you can do to measure protein folding. The one we're going to use is mass spectrometry, because it'll allow us to do this in complex mixtures across the whole proteome. So to do it, these are methods that have been developed in the Fitzgerald lab. We're going to introduce into the workflow a pulse proteolysis step, where we're going to use thermolysin, which can cleave at hydrophobic residues, to give a quick snap and cleavage of our proteins, and then we can read it out by mass spec. And if we do this as a function of denaturant concentration, the more a protein is unfolded, the more susceptible it will be to the thermolysin activity. So as we increase the urea concentration, we'll see an increase in the cleaved peptides. And if we do this and compare between an untreated condition and a treated condition, now we'll be able to see a difference in whether a ligand is bound or not. That change in stability will be a readout for that protein stabilization. So we're going to measure changes in protein stability, again, across the whole proteome to find proteins whose function is affected by these treatments. And we're doing this using a quantitative bottom-up proteomics readout, so we can measure thousands of proteins using this method. So back to the specific research question we were interested in, we wanted to understand the targets of our treatments and how we do them. So we're going to array them out as a function of copper concentration. So again, we treat with low copper and we get ICP-MS to get how much copper we can measure cell associated as a low concentration. We can treat with the ionophore itself, doesn't affect total copper concentration. But if we use our prochelator or the chelator, so PSEF-PT or PT, with the same amount of copper we've added here, you can see we get a huge boost in the total copper in the cell-associated copper. So our goal then is to generate both protein expression analysis by mass spec, proteomic mass spec, and stability profiles under these different conditions. And we want to identify the protein targets of copper toxicity. So again, we're doing two different experiments with the same treatments. One is a more standard bottom-up protein expression analysis, and the other is this STEP PP, the pulse proteolysis workflow that's going to give us stability. So these are the results just from treating with the low copper. And what you can see is that we get many more stability hits. So the highlighted ones are the ones that we calculate as hits, or we identify as hits, than we do with protein expression hits. But I want to point out that even at this small concentration of copper, you can see expression of COP-A. That's that copper transporter that I mentioned. So it recognizes that there's copper around and it's actively turned on. And QEO is that other protein involved in copper homeostasis or copper tolerance. So we can sense this level of copper, but it's not harmful to the cell. And across the other conditions, the thing to highlight is COP-A and QEO are also very highly expressed. We also see ZNTA, which is a zinc transporter, so that's interesting. But we see many more proteins where the stability is affected more than just their expression level. And that's what we find really interesting. So how do we define this now about what's driving these changes in protein stability? It could be copper, it could be the small molecule, it could be the metal complex that could be driving these stability changes. So we figured and reasoned that we could correlate this by how much total copper there is to figure out the targets of copper toxicity. And Nancy did a very clever cluster analysis to determine the driver of protein stability. So for example, this cluster of proteins, this is relative protease susceptibility, are very stabilized at the high copper, the conditions that cause high copper. So these proteins we identified as being a copper-driven cluster. And then we also have clusters that are driven by the prochelator, the cephalosporin, the small molecule, either stabilized or destabilized. And we've identified a lot of proteins, and what I want to focus on are the targets of copper toxicity here. Because one of the many classes of things that we found is we found many hits that are involved in central carbon metabolism, which we think is very interesting. So we see 3 proteins that were involved in the glycolysis part of the cycle, and another 3 protein hits that are in the TCA cycle. And conveniently, we found 2 proteins where we had convenient cell-based assays to test their activity. So one is this GAPTH, an early step in glycolysis. And these are whole cells being tested for GAPTH activity. So here's our baseline, and you can see as we're adding copper and pyrithione, we get inhibition of activity. IDH is another protein we identified. It's in the citric acid cycle, and you can see it also, its activity is inhibited. So these are proteins whose stability was shown to change by the proteomic method, and now we can correlate that with an inhibition at the assay level. We can also look at these proteins in vitro. So these are now purified enzymes where we see the inhibition of GAPDH as a function of copper. So copper inhibits GAPDH activity. And we can also look at changes in its structure. These data come from circular dichroism spectra. So you can see that copper inhibits the activity, but we actually get a stabilization of the fold. So the protein is more stable, but its activity is inhibited, which is interesting. On the other hand, IDH, again, purified enzyme, inhibited by copper. The data from looking at the circular dichroism spectra as a function of copper shows us that actually copper here causes a change in conformation. It alters its secondary structure. So two different kinds of outcomes where the stability is changed in different ways, but the activity nonetheless is important. So the function is clearly affected. So our results are interesting in the context of what other people have found for these pathways. So I'm showing here that what Hongxi Sun's lab found for silver toxicity, where they found GAPDH, and they were able to get a crystal structure where silver plus, silver 1, is coordinated at the active site cysteine and GAPDH, along with this histidine residue. Silver is often used as a surrogate of copper 1. So this is interesting to speculate, and we're working on trying to figure out more whether copper is inhibiting GAPDH in the same manner that silver does. And also this work by Kevin Waldron and staff aureus, looking at targets of copper toxicity, where they did a different kind of proteomic workflow, but under very high copper conditions, also saw changes in carbon metabolism and identified GAPDH as one of the targets in that system. Different organism, but now we're seeing some similarities. So to wrap up and leave you with some conclusions, what we found and others is that increasing cellular copper disrupts a wide range of protein targets. I highlighted a few that we've zeroed in on, but when we look at all of the hits we get from proteomics, we see a number of different proteins in different cellular processes that are affected by this increase in copper. We also hope to leave you with the concept that very clever chemistry, we can use that to direct this metal-induced toxicity, hopefully for selective antimicrobial activity. So designing molecules that are responsive to specific organisms or specific conditions of the immune system to be able to leverage this really powerful response that these microbes have to excess copper. We found that just looking at protein expression levels, or even I didn't show you any gene arrays, but even in looking at the gene arrays, we don't necessarily lead to identifying the targets of what the metal or the ligand is actually binding to cause those changes. I think of protein expression and changes in genome expression, transcriptions, as kind of the response of how the cell is trying to respond, but not the direct target. So we're really excited about using this method, and we're seeing that these changes in protein stability that we measure do correlate with changes in function, at least as we have been able to correlate with two of our hits so far, GAPDH and IDH. And these protein stability methods then can be very powerful tools for interrogating protein metal and more generally protein drug interactions. So I will end there, and I thank you for your attention, and I'd be happy to take questions if the format allows.
Thank you so much, Dr. Franz. I am waiting for some questions about the audience. Here is the Dr. Liliana Quintanar, so with a message for you. Great talk, and nice seeing you, Kathy. I was wondering about zinc TA expression of treatment with copper PT. What's going on? Have you measured total zinc? Can PT coordinate zinc, or is it specific for copper?
Oh, Liliana, these are great questions. I'm sorry I can't see you, but it's lovely to be here in Mexico with you virtually. ZNTA, this is a very interesting result, actually. We're very curious about it. So one, pyrithione is not copper specific. It will bind zinc. So we know that it can bind zinc, but we don't know what's driving the ZNTA changes. So what's interesting is that is actually driven by pyrithione, not by copper, at least in our cluster analysis. The ZNTA expression seems to be driven by the small molecule. So we're doing some experiments now to try to test hypotheses about what's going on there, and if pyrithione or its copper complex or its zinc complex does something to block ZNTA in some way. I mean, it could. We saw in another set of experiments I didn't talk about, but looking, in this case, we were looking at membrane protein knockouts for what would be affected. And the ZNTA knockout on E. coli is highly susceptible to pyrithione toxicity. So we think that there is something really interesting here. We don't know if it's acting on ZNTA because of the copper, because of the zinc, or because this small molecule can maybe coordinate the sulfur or do something that would inhibit ZNTA or cause some change. So we have a couple of hypotheses we're testing, but it's interesting. You saw that very interesting result in there. So thanks for pointing that out.
Thank you so much for your answer. Eugenia Luisa Silva have a questions. Why do you expect to find selective antimicrobial activity if the target seems to be general carbon metabolism? Have you found any species or genus-specific targets?
So another really great question. So bacteria are very sensitive to copper. So if you, actually cells are in general. So we think this is something very primal about why proteins don't like to have excess copper. So targeting that specifically would be very challenging, to say, oh, only in a pathogen cell I could have this copper toxicity, or that's a challenging thing. So the way that we're going about that is to design molecules that are responsive to something that it can be specific. So in our example I talked about today with PSEF-PT, that molecule is selectively released by the activity of this drug-resistant enzyme. So if it's just a commensal organism that isn't posing harm to the host, then the molecule doesn't get turned on. So we don't release this pyrithione molecule that has a more generic response, right? It would be toxic to whatever cell. But if we have it be selectively turned on, that's kind of the angle that we're trying to use what is a very general toxic mechanism and focus it to something specific. At least that's our approach so far. It's a very good question. Thank you.
Thank you so much. I have a question for you. In this context of methyl ions enter in the cell, the transport of copper change the function is more present in the cellular membrane, or change something to introduce more copper in this environment?
So is your question, is copper localized in like the periplasm or in the cytosol?
In the cell salt.
So in at least in E. coli, the periplasmic space is actually the space of copper resistance. So copper becomes toxic when it's in the cytosol. So that copper exporter, COP-A, that we identified and we know is important for copper resistance, that's on the inner membrane, inner membrane part. So it pumps copper out of the cytosol. But in the periplasm, copper can actually accumulate to fairly high levels. So total copper can actually be quite high. And there are even some bacteria that can accumulate so much copper in their periplasmic space, they look blue because of so much copper. So it's really not total copper that causes toxicity. It's copper that's not controlled in its right place. So the periplasm, there are these storage proteins and QEO that can accommodate this extra copper where it's not toxic. But if it slips into the cytoplasm, no good. I don't, does that answer your question, Trinidad?
Yes, thank you. Okay, we can have more questions. Then I want to thank again because to accept this invitation and be here with us virtually as the National Institute of Genomic Medicine. Thank you so much, and it was a pleasure see you again.
Thank you, my pleasure, and hope to welcome some of you to Durham and North Carolina someday.